genetic diseases and inheritance pattern, autosomal dominant condition. The main objectives are to understand the basic concept of genetic diseases, to know about the inheritance pattern, to explain the autosomal inheritance patterns, to know about the autosomal inherited disorders. Introduction. The pedigree analysis is used to study the inheritance pattern of a gene. Based on family history, the particular trait is recorded and drawn systematically in the pedigree chart. The chances of occurrence of that trait can be estimated, and the reasons for this type of inheritance pattern can be unraveled. A proband is an affected individual shows the genetic disorder in a family for the first time. When you look at a gene, it will have two copies. One is inheriting from the mother and another from the father. Any changes that occur in the gene will lead to variations. These variations in the human genome make them phenotypically different from the other individuals. Some of the changes termed as mutations, and they can alter the functions and cause genetic diseases. The mutation of the gene still exists even if get affected by the mutation. The mutation may affect any one copy of the gene, while other copy can function correctly. One copy of the altered gene is enough to cause the disease, which is the dominant condition. The one dominant affected copy of the gene will mask the function of the other copy of the gene. Genetic diseases can occur due to the mutation that affect the function of the protein formed. Genetic disorders can be inherited if the mutations occur in the germ cells. The parents can pass the mutation to the offspring through the germ cells, whereas the genetic diseases can also be happened by the mutation in the body cells or somatic cells. Some of these diseases can be Mendelian diseases, where the mutation occur in a single gene. Some mutations can occur in multiple genes by the influence of some environmental factors, are termed as multifactorial genetic diseases. Hence, this episode explained further in the following subheadings: A. Autosomes and the inheritance. B. Autosomal dominant inheritance. C. Autosomal dominant disorders. Autosomes and the inheritance. Autosomes are the non-sex chromosomes. Humans have 22 pairs of autosomes and 1 pair of sex chromosome. The genes which are present in any of the autosomes get inherited to the next generation, then it is termed as the autosomal inheritance. Autosomal inheritance of disease will affect both male and female child equally. If the father has one altered copy of gene capital A small a, capital A indicates the normal copy and the small a shows the affected one, and mother also has one affected gene capital A small a, then the possibility of getting children are capital A capital A, capital A small a, capital A small a and small a small a. In the case of recessive condition of inheritance, both the copies of the gene should be affected, that is small a, to show the symptoms of the disease. Children with one copy of the altered gene, capital A, small a, are called the carriers. They carry those genes and inherit that affected gene in the next generation. Hence, chances of getting affected child are 25%. In the case of autosomal dominant inheritance, one copy of the affected gene is enough to get the symptoms of the disease. By taking above given situation, both father and mother will be in altered condition with the genotype capital A, small a. Then the children with genotypes capital A, small a and small a are affected, and the children with capital A and capital A genotype are normal. A single copy of the gene is sufficient to develop the symptoms of the disease. In this case, 50% of the chances occur to get the affected children. B. Autosomal dominant inheritance. In the case of autosomal dominant disease, the mutated gene will present in the autosomes. One copy of the mutated dominant gene is enough to develop the disease in affected individual. If a man with affected gene marries normal woman, then there will be 50% chances of getting children with affected genes and 50% of children will be normal. The mutated gene encodes the protein which are usually the structural proteins. Defects in the structural protein can occur if any one copy of the gene gets affected. In contrast, the defected enzymes are formed by the mutated gene with two affected copies of alleles. The autosomal dominant condition can be easily identified in a pedigree. When a pedigree is drawn based on the family history of affected individual, the traits can be easily identified by their inheritance pattern. Since it is a dominant condition, the trait will appear in every generation. The dominant condition affects individuals in every generation. The changes in the gene are dominant and it can mask the normal function of other copy of the gene. One affected parent is enough to get the affected children. In a dominant condition, one copy of the altered gene is enough to show the phenotypic change. Offsprings with one modified copy of the gene will develop the symptoms. There will be no skipping of generation. In each generation, the affected one copy gene gets inherited to any of the offspring and show the phenotype of the developed disorder. In this condition, both male and female child get equally affected. Dominant condition of the autosome gene will show up the phenotype both in male and female. The chances of getting the disease are 50% in the offspring. Half of the autosomes from father and another half from the mother get inherited to children. Individuals with both normal copies of genes will not pass any affected trait to their offspring. Some of the examples of autosomal dominant disorders are neurofibromatosis, Huntington disease and Marfan syndrome, etc. See examples of autosomal dominant disorders. 1. Neurofibromatosis. There are many different disorders which get inherited in the autosomal dominant condition. One such disease is the neurofibromatosis. This genetic disorder mainly affects the nervous system. The mutation in a gene of nerve cell leads to the altered functions. There will be the formation of tumors all over the body, and tumors can be benign, but in some severe cases they can be malignant. There are mainly two types of neurofibromatosis, and they are neurofibromatosis 1, that is NF1, is caused by the mutation in the gene NF1. This gene is located in chromosome 17. This gene codes for a protein called as the neurofibromin. The normal function of this gene product is to maintain the normal proliferation and growth of the cells. It also can stop the overgrowth of some cells called nerve sheath cells, which helps in support of the nerves. Hence, NF1 acts as a tumor suppressor gene. When the mutation occurs in the gene NF1, it codes for the protein showing abnormal function. This leads to the proliferation of cells and the formation of tumors in the nerves, in skin and other parts of the body. There is also the formation of dark pigmented patches on the body. NF1 can be caused when the affected gene inherited from parents to offspring. It can also be caused by some of the spontaneous mutation in the gene. NF1 is a common type of neurofibromatosis. Neurofibromatosis 2, NF2. Gene NF2 is located in chromosome 22. It is also a tumor suppressor gene and encodes for a protein called Merlin. This protein will regulate cell growth by interacting with many other proteins. When the mutation occurs in the gene NF2, the formed Merlin protein shows the abnormal function. The formation of tumors occurs in the spinal, cranial and auditory nerves. The tumors in the auditory nerves will lead to the hearing loss in the early young age. NF2 is the less common type of neurofibromatosis. Other symptoms of neurofibromatosis include hypertension, high blood pressure level, macrocephaly, large size of the head than normal size, short stature of the body, learning disability, abnormal spinal structure, curvature in the spine, bones will be enlarged and will not function properly, cataract, problems in balancing the body. This disorder is relatively common and the frequency is approximately 1 in every 2000 to 3000 new births. The incidence of NF1 was found to be 30 cases per 1 lakh new birth in Germany. Diagnosis and treatment. This disorder can be diagnosed by several tests like medical history, family history, physical examination, etc. Doctors will ensure that whether the same disease present in the family of the affected individual. Several physical studies and scanning of the body are done to check the symptoms. Magnetic resonance imaging and X-ray scanning can be done. Some of the simple hearing balance tests are also conducted to check the state of the individual. There is no cure for this disorder. The only possibilities are to avoid the symptoms of this disorder. The developed tumors can be removed by surgery and other techniques like chemotherapy and radiotherapy. The pigmented patches will be in the body without any treatment in most cases. The symptoms of neurofibromatosis will be in mild condition. Only some of the cases will turn into severe and need medications. It also depends on the type of neurofibromatosis that occurs. 2. Huntington disease. It is an inherited condition that mainly affects the nerve cells in the brain. It will create the imbalance in every function, including body movements, mental problems. The defect occurs in the gene called HTT. This gene is located on the short arm of the chromosome 4. This gene is responsible for the production of a protein called huntingtin. This protein plays a role in the nerve cells. The defect in the gene HTT will result in the formation of abnormal protein which causes the Huntington disease. The gene involves the sequence which consists of CAG, cytosine, adenine and guanine. Repeats. The normal condition shows about 10 to 35 repeats of this sequence in the gene. The affected individual will show more than 36 repeats in the mutated HTT. These repeats can be up to 120 times in some cases. Some of the symptoms of Huntington disease include mental problems, difficulties in learning things and decision making, lack of coordination, slow movements, mentally depressed conditions and irritabilities. Huntington disease occurs typically equally in both gender and it gets start in mid age of life, that is 30 to 45 years. But the onset of disease can also find an early age. In the case of children, it is not possible to live till adulthood. The frequency of this disease is occurring about 5 to 7 per 1 lakh in some Western countries. 3. Marfan syndromes. This condition leads to disorders which mainly affects the connective tissues. The gene affected in this condition is FBN1. This gene is located in the long arm of chromosome 15. This gene encodes for a protein termed as the fibrillin 1. This protein will bind with many other proteins to form a structure called microfibrils. These structures are the unit of fibers and they strengthen the connective tissues. By binding with growth factors, microfibrils will help in the growth of tissues by releasing those factors when that is necessary during the repair. Mutation in the FBN1 will lead to the formation of the protein with abnormal function. This will lead to the reduced formation of microfibrils. Lack of microfibrils further leads to the uncontrolled growth of the tissues, and then they lose their flexibility. The instability of the tissue takes place and organs of the body get affected, including the heart, eyes and bones, etc. in the Marfan syndrome. Some of the phenotypic symptoms of Marfan syndrome includes affected individual will be tall and skinny. They possess long limbs and fingers and toes will be elongated, abnormal spinal system, abnormal shape of chest development. They can be in sunken or protruding condition and narrow face with abnormal teeth. The prevalence of this condition is rare in India. It has been estimated that 1 in 5,000 to 10,000 people live in affected by this Marfan syndrome in the general population. The autosomal inheritance shows the inheritance of genes located on the autosomes. In the case of the dominant condition, one copy of the gene is sufficient to express the phenotype of the disease condition during the heterozygous state. Thank you.